Welcome to Study Loop.
We are being asked to determine which statement is false about epigenetic regulation. So first, let's see what the term epigenetic means. The prefix epi means above. So epigenetic regulation is regulation above the gene. What does that mean? Shown in here in this diagram is it's a double strand of DNA and how the DNA eventually wraps around histones. Histones is a group of 8 proteins. So histones are actually a protein, and 8 histones together make that spherical structure around which DNA wraps, and that is how DNA is organized in the cell. Now, to DNA, you can add methyl groups, and the addition of methyl groups can affect the affinity of the DNA for transcription factors, which then changes how often that stretch of DNA is transcribed. Or to the histone tails, you can add acetyl groups, which affects how tightly the nucleosome. So DNA plus histone complex is called a nucleosome. And that's one of these guys right here. Okay, that's a nucleosome. The DNA with the histone complex together. So the addition of acetyl groups to histone tails affects how closely packed those nucleosomes are. How does that happen? So, and on the left side here, so let me switch to the color of the pen. On the left side, on this side, we are showing how the addition of acetyl groups, so over here, going from top to bottom, the addition of acetyl groups results in the exposure of more DNA. And if this DNA is exposed, it can be transcribed. How does this happen? So on the left side here, that's what we're showing. Histones are usually acetylated at lysine residues. Lysine residues are generally positive in the cell. When you add an acetyl group to that lysine residue, you are removing that positive charge away. This matters because DNA is negatively charged because of the phosphates in the backbone of the DNA. So if the DNA is positive and the lysine, if the DNA is negative and the lysine is positive, there is an attractive force here. But when you take that positive uh, charge away from the lysine, the DNA doesn't feel as strong of an interaction with the lysine. So that's how the acetylation of histone tails results in a looser chromatin structure. Okay, so choice A is false. Histone protein does not become more positive, it becomes less negative. This means that the rest of them must be true. So let's quickly look at the rest of the answers. DNA molecules are modified within CPG islands. What are CPG islands? So what I'm showing here is a representation of a stretch of DNA, and shown in the dashes here is just your gene. It doesn't matter what it is, it's just some gene. Okay. Now CPG islands, which I've drawn here, they are part of the promoter. Okay, and CPG islands are called that because it is a DNA sequence that has a lot of repeats of C and G. It's CG, CG, CG. The P in the middle refers to the phosphate that's in between nucleotides. So you can also alternatively call these guys CG islands, sort of the same thing. And CG islands are important because this is where the DNA is methylated. So when we're talking about epigenetic control, this is where it happens usually. It's in this CPG islands. So choice B is correct. DNA molecules are modified within CPG islands. Choice C, methylation of DNA and histones causes nucleosomes to pack tightly together. Now, this choice is a little bit difficult to absolutely 100% get behind. And the reason that I say that is because methylation of histones can actually cause both looser packing or tighter packing of the histone. And we don't really yet know the mechanism of how that happens. So if you notice here, the addition of the acetyl group, which is hydrophobic, resulted in a looser structure. Methyl, so let me pick a different color here. Methyl is also hydrophobic. Okay, so methyl is a C with an H with an H and another H. So if you add that in here, that's also going to make the lysine less positive. So it could have the same effect as acetylation. However, the methyl group could be added to a different kind of residue. And we don't really know. So the methylation experimentally has been observed to both loosen or tighten the chromatin structure. DNA methylation always makes it more tight. Now, we have observed this experimentally, but we have not determined the mechanism. So choice C is sort of like 75% true. Okay? Choice D, histone acetylation results in the loose packing of nucleosomes. Yes, that's exactly what we're showing right here. The addition of, so right here, the addition of acetyl groups results in a looser packing chromatin structure. Okay, but choice A is the one that is absolutely categorically correct. And remember, when you're answering questions, choose the answer that is the most correct. And in this instance, that's choice A.